So when we look at the cell wall, it's made of something called peptidoglycan. So if you think about the name, remember peptide indicates proteins. Glycan, think of glycogen, one of the polysaccharides that we talked about. So this is really a molecule that's long chains of carbohydrates connected by short chains of amino acids. And when we look at plant cells, we'll see that they have a different type of material we've already talked about a little bit, called cellulose, that makes up their cell walls. And fungi, like mushrooms, have cell walls, but they have a different type of chemical called chitin. So even when we see the same types of structures in cells, they aren't always made of the same types of materials. Now, what does the cell wall do? Keep in mind what I said, it's very thick, it's very stiff. So the cell wall has a protective function. What does it protect the cell from? Physical forces. A lot of bacteria can move. Well, anytime you move, there is friction. So when we look at this friction, that can eventually wear the edges of the cell down. That's basically what happens to your tires and why you have to get new tires every once in a while. So in this case, the cell wall is what gets worn down, not the plasma membrane, which is very soft and would wear down quicker. And actually the cell wall isn't alive. So if the cell wall gets replaced before it's completely destroyed, yeah, no sweat. It also protects bacteria from certain antibiotics. This is how one antibiotic actually works. There's an antibiotic called penicillin. And what penicillin does is penicillin disrupts the cell wall of bacteria. And when it disrupts the cell wall, it either kills them or they at least have to stop growing. So your immune system has a chance to take care of them. And finally, it also stops bacteria cells from absorbing too much water. One problem that a lot of cells have is that they can absorb so much water that they burst like a balloon that you're blowing up too much air into. But when a bacteria cell starts to absorb water, it starts to swell. And as it swells, that cytoplasm and plasma membrane that presses out against the inside of the cell wall. And the cell wall is so thick and stiff and strong that after a while, the cell just can't absorb any more water because it can't push out any further. Now, as we'll see later, human cells don't have a cell wall, so human cells can actually absorb so much water that they burst like a balloon. Now, a lot of cells outside the cell wall, they have a third layer called the capsule, or sometimes it's even called a slime capsule. And this is made of a chemical called a mucopolysaccharide. So polysaccharide, that's a carbohydrate. Muco refers to the fact that it's got a chemical called mucin, mucus in it. And it does feel kind of slimy. And like the cell wall, the capsule provides some protection for the bacteria cell. It's hard for things to get through that slimy outer layer to get into the inner parts of the cell and damage it. Because remember what we said, cells are factories that make proteins. Well, all the protein making machinery is in here, the cytoplasm, the DNA, the ribosomes. As long as we keep them safe, the cell is good. Now, some bacteria cells have long strands that stick out past the cell. So some bacteria cells have a flagellum, or some have more than one. And in bacteria cells, this flagellum twirls around, kind of like the propeller on a boat engine. And when it twirls around, the bacteria swims through its environment. Now, keep in mind, bacteria are really small, so when we say they swim through their environment, it takes them maybe 2 or 3 days to swim an 1/8 of an inch or a 1/16 of an inch, so they don't swim very quickly. The other type of projection that we see sticking out of some bacteria is a much shorter structure called a pilus. And a pilus attaches a bacteria cell to a couple of different things. When bacteria cells exchange DNA, now they don't reproduce sexually, I said earlier, but they do exchange some DNA. We'll talk about this later. When they exchange DNA, they can use this pilus to attach to each other so that, you know, this transfer is stable. If they're a bacteria cell that causes disease that attaches to, say, a human cell, they can use this pilus to attach to one of your cells, to what we call a host cell. And not only can they attach to other cells, but they can attach to non-living material. So the bacteria that cause dental caries, what we usually call cavities, the bacteria that cause that can use their pilus to attach to dentin and enamel on your teeth. Microbiologists sometimes give each type of pilus a different name depending on whether it attaches to another bacteria cell in DNA exchange, whether it attaches to a host cell that's being infected, or whether it attaches to what's called the substrate, something that the bacteria cell lives on, like the enamel of your tooth. Sometimes they all get different names. We're just going to call it a pilus. You can take microbiology to learn all the different individual names if you're interested. So when we look at a prokaryotic cell, it's a pretty simple way to make proteins. And it's very easy to get the proteins where we want. If we look at this whole prokaryotic cell, let's come down and look at a eukaryotic cell for a second. So here's a eukaryotic cell. If we look at this little blue thing here, or there's one over here that's kind of cut open, this is a mitochondrion. A mitochondrion is about the size of a really, really large prokaryotic cell. So notice prokaryotic cells are much smaller than eukaryotic cells. So one of the things this means is when we look at making proteins, we never have very far to transport the protein to where we're going to use it. So we don't have any special transportation system. Now, in a eukaryotic cell, because of the fact that the cell is so big, these little dots here represent our ribosomes. We could be making a protein here, but we might want to use the protein way over here. So we're going to have a very well organized transportation system to get the proteins to where they're finally located. Okay, are there any questions about prokaryotic cells? And what I will do is I'll go back and I'll check and see if I have any recordings from a prior semester that I can upload. So I'll put up the last part of class from today, the recording that we did at the end. But I'll see if I can add that other recording. So if you want to check the YouTube channel later today, it may take me a while to locate it. But I'll see if I can find it, and if I can, I'll upload it. So are there any questions? Okay, if not, we'll stop screen sharing. Don't forget, shortly after class, I will have that homework assignment that's due Monday morning before class. That will be up. Now, there are some drawings of molecules in that homework assignment. So I'm posting it as a Word document so that you can type on it directly. But I'm also posting a PDF version. So if you don't see the actual molecules showing the bonds, if you don't see that diagram in the work document, check the PDF document. And then if you can't see it on that, let me know and we'll try plan C. Any questions before we stop for the day?